Every day, thousands of patients, researchers, and clinical staff cycle through the corridors of the Herston Health Precinct. It is a massive, continuous flow of human traffic passing through one of the most concentrated medical hubs in the country. Managing a facility of this scale requires a distinct break from standard commercial cleaning. Wiping away visible dirt and polishing floors will not suffice here. Healthcare decontamination is an exercise in total microbiological eradication. Without intervention, high-touch surfaces like stainless steel bed rails, light switches, and door handles become transmission highways. A pathogen introduced by a single touch can migrate across a ward in hours. At the microscopic level, these organisms multiply exponentially. They establish colonies on sterile surfaces, overwhelming the immediate environment, preparing to spread to the next host. Halting this progression requires a specialized, invisible workforce. These crews execute highly regimented, scientifically validated protocols to break the chain of infection before it reaches vulnerable patients. A single failure in surface decontamination creates a dangerous vector. One missed protocol can trigger a hospital-acquired infection, halt critical medical research, or cost a patient their life. The first zone of containment operates on the front line of daily medical care, the general patient wards. Even with TGA-approved hospital-grade disinfectants, pathogens do not die the moment a chemical hits the surface. Eradication demands strict adherence to contact time, the exact duration a surface must remain wet with a specific chemical to achieve a verified kill rate. Quaternary ammonium compounds require 10 minutes of wet exposure to eliminate common healthcare pathogens, while accelerated hydrogen peroxide completes the task in 1 minute. As the 1-minute threshold crosses, the chemical degrades the cellular walls until nothing viable remains. High-turnover surgical areas within the Royal Brisbane and Women's Hospital deploy these faster-acting agents to clean operating rooms. The reduced contact time keeps patient flow moving quickly without compromising the sterile field. This chemical application relies on dedicated physical tools. Color-coded microfiber technology traps organic material, and strict segregation protocols ensure clinical waste never crosses paths with sterile supplies. Synchronizing the correct chemical agent with its required contact time forms the first vital barrier against multi-drug-resistant organisms. When a patient is discharged, the room undergoes a terminal clean. Every surface, fixture, and piece of equipment is broken down and systematically decontaminated to return the space to a baseline sterile state. In critical care units, visual inspections are inadequate. True biological cleanliness must be scientifically measured and verified. Crews swab high-touch surfaces to collect invisible traces of cellular material, specifically looking for ATP, the energy molecule found in all living cells, including bacteria. This readout shows a luminometer converting that biological swab into an exact quantifiable safety score, instantly revealing whether microscopic biological matter remains on the surface. The requirements escalate further inside ISO 14644 cleanrooms. Here, the threat expands from the surfaces to the ambient air itself. These rooms maintain positive atmospheric pressure, pushing air outward so contaminants cannot drift in. Simultaneously, continuous HVAC HEPA filtration physically scrubs microscopic particulate matter out of the indoor air. By utilizing ATP swabs and calibrated particle counters, the subjective task of wiping down a room transforms into a precise, data-driven medical science. The most stringent protocols operate deep inside the precinct's highest risk zones, the QIMR Berghofer Biomedical Research Laboratories, which include biosafety Level 3 containment facilities. These environments handle serious airborne human pathogens, including strains like Mycobacterium tuberculosis. Researchers operate in heavy personal protective equipment, confining their active biological work entirely within the boundaries of sealed biosafety cabinets. Personnel follow severe entry and exit restrictions. Leaving the containment area requires mandatory decontamination showers, and every piece of protective gear, wipe, and chemical container used during cleaning is immediately classified as biohazard waste. While cleanrooms push air out to protect a sensitive environment, BSL-3 labs operate in reverse. They use strict negative pressure to pull air inward, ensuring hazardous materials cannot escape into corridors. This cross-section schematic illustrates negative pressure, with airflow vectors pointing strictly inward to trap airborne pathogens. Before waste leaves, it undergoes thermal sterilization inside an industrial autoclave, baking at exactly 121 degrees Celsius for 30 minutes to destroy all biological agents. These overlapping layers of inward airflow, chemical eradication, and intense thermal sterilization form the ultimate failsafe, keeping hazardous pathogens isolated from the broader Brisbane community. The Precinct's defense system is entirely interconnected, from a 1-minute chemical spray applied in a busy surgical ward to a 121-degree autoclave cycle burning in a subterranean research lab. Positive air pressure, rigorous surface toxicity testing, and total biohazard containment operate simultaneously, 24 hours a day. Together, they project an unbroken, invisible shield over the Herston precinct. Beneath the relentless pace of BLE clinical care, this microbial defense grid holds steady. Doctors and scientists perform the visible work of diagnosing illnesses and pioneering medical breakthroughs. Yet their ability to work safely depends entirely on this hidden foundation of continuous infection control. By executing these decontamination protocols without compromise, this invisible workforce secures the precinct's operational continuity, protects patient health, and maintains public trust.